next speaker is Orla Doyle. She joined the Novartis Statistical Engineering Group 2 years ago, and she is going to present an R package and Shiny app developed by Novartis to facilitate the estimation of probability of success of a clinical program.
Indeed, thank you very much. Great to be here with you today. I think this is one of my first in-person presentations since the pandemic, so really nice to see some faces, and so far everyone looks awake, so hoping I can keep that success for the next 10 minutes. So I'm going to take a little bit of a different approach today and not just talk about the technical details, but how we worked together, how we collaborated, and the things that we did to try and do this rapidly, but also in a very sustainable way. As there are my disclaimers. But first of all, let's think a little bit, just so we have the context for what we're talking about today. So when we talk about probability of success in this context, we're talking about the probability of success of a trial. So as many of us are familiar, we have a lot of very important decisions to make during the drug development process. And as much as we can, how can we be evidence-based? How can we be data-driven in those decisions? And POS gives us an additional data feed to help with that decision-making process. So certainly not the only piece of data, although we like to think of ourselves as very important. There's many other factors that go into it as well. And typically, where we use this type of methodology today is in this transition from phase 2 to phase 3. And that's kind of what we'll focus on as we go through it. I'd also like to acknowledge many of my very clever colleagues who were involved in developing this methodology. This has been published as well, so you can read the paper. I think it took me about 10 different goes at reading it before I actually truly started to understand it. So quite a lot of complex Bayesian modeling behind this as well. And I'll try to explain it as succinctly as I can, while also giving you a feel for it. So as Francois really nicely gave us the kind of some of the concepts behind Bayesian modeling, here what we're trying to do is start at what do we know before we even begin? What might we know from the industry? So in step 1 here, we're taking proprietary data and we're actually using kind of this industry-wide view of what is happening with quite crude metrics about our study and our product. So very high-level characteristics about, you know, what is the molecule, what is the therapy area. So lots of things that we know readily when we begin. And then that gives us our first industry benchmark. And this is what we often see across many companies when we measure probability of success. What we do a bit differently here is as we move into the later steps, we start to bring in what do we know from what we've observed so far. So for example, what have we seen in our phase 2 trials or even phase 1 trials? What have we seen in terms of that's giving us additional evidence or information about the effect size, about how we might be thinking about something called our target product profile, which is kind of what we're trying to aim to get to during drug development. And the industry benchmark here is really working as the prior, so it's calibrating us in terms of should we be pessimistic, should we be optimistic. And then the evidence that we have seen already is kind of our central model here. But as we move through it, there might be things that we know that are hard to quantify. They might be more qualitative, and that's where step 3 comes in. Here we start to think about, are you worried about alignment with the regulator? Do you think this is going to be a difficult product to develop technically? So we start to bring in these more qualitative sense of risk that we might have here as well. And ultimately at the end, we're trying to bring forward one single number to quantify all of these very complex and very different types of risk or success, however you want to see it. And then, of course, in exceptional cases, there might be something very subjective that, again, doesn't really fit into any of these steps already, so we can invite people to further calibrate what they're doing. So really exciting, gives us lots of information, distills a lot of things down into one number that's quite, understandable for people from very different backgrounds. So where I started on the project and where I came in is we already had a prototype app developed in OR. This was in Shiny. And we wanted to know, okay, how do we take this so that we can scale it, so that it's maintainable, and how can we bring in some of these best practices in terms of programming, right? So unit tests, functional tests as we're looking at the app. How can we kind of elevate it a little bit further, given the importance of what we're measuring? We also talked to users and they said, I really want a bookmark. I want to save and restore. I want to have a project management function within this app. So before we started, we wanted to really understand who were we serving and what did they need. I think this is one of the few times in my life where people were super clear about what they wanted. So it was like, okay, we want save and restore. That's the most important thing that we want for us. I'm so tired of having to redo some of my things in this prototype. And that was really brilliant to have that focus, because then we could be very disciplined as we went through the development. But the one thing that we learned as well is some people want a UI, some people don't. Some people only trust something if they're in the detail, if they're in the code. Some people are like, I don't have time for that. I want the UI. I'm happy with it. So how can we serve these two groups? Additionally, as I mentioned, it's a complex methodology, right? We've tried to cover as many use cases as we can, but certainly there's probably things that still fall outside the framework. If we enable users with the code, with the backend package, hopefully they can kind of join us, iterate, and then we can serve even more use cases as we go through it. So who were we? So we developed a team. So I was part of the back-end team with Andrew and Marcus, my colleagues. And then on the front end, we have really great Shiny experts as well in David and Mustafa. And we really formed one core team between us. It was really important for us to have really good communication. We had so many dependencies between what we were doing in the back end and the front end that we really needed to go together on this. So making sure that we had really good communication and really good planning for what we were doing. Also really good connection to our users. You know, how were we talking to them? How did we want to talk to them in terms of the forum, in terms of when we would demo to them? And as I mentioned earlier, we have a whole bunch of subject matter experts who know this methodology inside out. You know, often we would want to kind of run things by them to make sure that we really understood what it is that we were doing. And we started with lots of diagrams, and then those diagrams got uglier and got more detailed, but I'll show you the nice ones for now. So really simply, when we started, we wanted to separate the package from the Shiny app. We wanted to build a database. So really at a simple level, what did that look like, and what did the handovers and connections need to be in this really more simple world? As we drilled into it, we wanted people to log into the app. Now, this is an app where you're talking about the success of a clinical trial. So we knew we would need to think very carefully about the privacy or the sharing permissions of the different projects. So maybe 2 people wanted to share, but they didn't want to share it with everyone. Maybe we wanted some examples to be public. So what did all those permissions need to look like as we moved through it? So as I said, I've only showed the nicer diagrams here. There's spider webs of ones as we got deeper into it. The one thing that we did that I would highly recommend anyone to do, which saved us so much time, is we went to a wireframe or a mock-up and we drew what we thought the app might need to look like. This is using a tool called Balsamic, and we just drew it. There's no code behind this, but it feels and looks like code to a certain level. And we showed it to people, and they were intuitively like, I don't really like that the share button is up there. I wouldn't have thought that should be there. What did you mean when you said settings? All of these things. So we got a lot of the feedback up front before we started to write the code. And on the left-hand side here is what the app that's launched today actually looks like. I don't think it looks terribly different, which is good. But as I said, we did get a lot of that really kind of sticky feedback at the start that I think would have been really inefficient for us to rework afterwards. We then also did the back end in an object-oriented approach. And at the time, it feels more fun to start coding. It took a bit of discipline to actually stick with this. So to actually sit down and map, what objects do I need? How do they depend on each other? How do they talk to each other? What are my fields, my attributes, and what are my methods? So again, this took a little bit of discipline to kind of stick with this and see it through. It was much more tempting to go to the code. But also this gave us a great framework for the testing. So each of these classes then can have their own suite of tests. We can go to the previous app, we can go to our SMEs and say, what should this look like? We could also build adversarial tests, so things that we knew should break it, and build a lot of those exceptions and things in. There's a lot of functionality that I haven't showed here specifically that kind of brings everything together, but I think this is really the meat of it. And this gave us a lot of very natural structure in what we were doing. And then we did a lot of classical stuff, managing our environment using Renv, test that for the unit tests and things like that. So overall, and then we built all of this into our CI/CD. So every time I merged, I was to cross my fingers and be like, this is going to work. Sometimes it did, sometimes it didn't, and I knew straight away. Sometimes, and as we got more and more tests, you can see sometimes here, this is taking just about enough time to go and make a coffee, so you can have a little break and come back, and you can keep your fingers crossed that whole time. So that kind of really helped us, right? So before I even said, can I get a code review, I knew whether things were breaking or not, which was really good. We invested a lot in our discipline in using tools like Jira and GitLab. Again, this was a different way of working for us to begin with, but it helped us to keep our meeting load lean. So you don't need to say, hey, what are you working on today? Because I need you to test this. You could see what everyone is working on before you even ping them, because we have our tickets and you have everything in progress and you have your comments. So I think that really helped us as well. Wireframing. You know, I said that earlier, like that really helped us to be efficient, and investing in the tests. So sitting down and saying, what would it look like if I put these parameters in and I get this out? Investing in that upfront gave us a lot of tests then that we could use through our development. And then we did a lot more with packages that are developed by David Grandjean, like Shiny Validator as well. And then in the end, separating the front end and back end for different user groups. So very quickly, I'll just talk about the open sourcing of this. So right now we're in a period of hypercare. We just launched this in April, so we're just trying to make sure that everyone gets served for their needs. But we are working towards open sourcing this package as well. Just to note that the benchmark will be BYO benchmark, just because that's on proprietary data that we buy from a vendor. So that's the only wrinkle at this moment in time. But thank you so much, and a pleasure to be here today.
Thank you. Next talk is going to be given by Martin. Martin works at the strategic consulting group of Cytel, and he will present Shiny app, which allows to continuously monitor a trial and determine if recruitment is sufficient.
Yeah, it works great. Okay. Yeah, thank you for the introduction. Now works. Okay, great. Yeah, this is application that is co-developed with Ursula, who will later be on the panel discussion. So we call it MAST, Goldilocks approach for selecting the final sample size. So the motivation of this, just a quick overview of the talk, and you see the usual disclaimer. Another disclaimer that nowadays seems to be necessary: we didn't use ChatGPT for anything in this application or for the talk. So, the starting point for this was a publication from Orla Doyle, who introduced the Goldilocks principle into clinical trials. You probably all know the fairy tale, Goldilocks and the 3 bears. So in clinical trial, this is constantly asking, is the sample size too big, too small, or just right? By using a Bayesian framework and regular interim analysis, assessing the probability of success for the end of the trial. So this uses unmasked data. Further inspiration we took from a publication from Xi and colleagues who proposed a dynamic monitoring with real-time data. And this again uses unmasked data. So our idea was to use dynamic monitoring of a nuisance parameter throughout a clinical trial for blinded sample size re-estimation. So the naming of this approach or application was relatively straightforward. So masked Goldilocks. And it's also relatively easy to find a logo. So please welcome our superhero, masked Goldilocks. Maybe a quick introduction or reminder, what is blinded sample size re-estimation? It's around since a long time, and is mentioned here as seen in the ICH A9 guideline from '98. So when you have a longer running trial, you usually have the possibility to check your initial assumptions that were used for the sample size calculation at an interim analysis based on blinded data. And then you can use these blinded data to assess, for example, the variance or event rate in survival analysis, and see if this is as anticipated or not. And then do a revised sample size and document this in a protocol amendment. Later, the FDA guideline assessed or specified that this probably should be pre-specified. But again, it's the method is around a long time, and it's recommended. And but is it actually used? And we did a literature research for the term blinded sample size re-estimation, and astonishingly, it's not so much documented. Maybe it's used, but at least it's not very often mentioned in the publication that a blinded sample size re-estimation was used. So that brought us to the idea, is it actually used, and is it easily accessible? Let's go a little bit to the practice. So here you see sample size calculation or specification in the protocol. Let's take, for example, an endpoint that is changed from baseline, and the analysis is an ANCOVA adjusted for the baseline plus maybe some covariates. And the typical text for the sample size calculation for a clinical protocol, trial protocol, could be a total sample size of X patients, ensures a power of at least 90% to detect a difference of Y between the group with a standard deviation of Z using two-sided 5% significance level. So at first sight, this looks straightforward and relatively clear. But then at second thought is, what is actually the standard deviation? Where this comes from? There are three possibilities. Could be the standard deviation of your outcome variable, could be the standard deviation of the change from baseline, or it could be some kind of adjusted standard deviation back calculated from the standard error of the LS means of the treatment difference. And so it's sometimes it is not clearly documented where the standard deviation comes from. And this matters actually, because when we found a nice publication where this is documented, where this is illustrated, what sample size can vary, the estimated sample size can vary in function of the correlation between the baseline and your at-visit value. Obviously, if you only use the at-visit value, the sample size doesn't change, because here, in function of the correlation, there's no change. So this is set to 100. But when you use instead the change from baseline, and you have a very low correlation between the baseline and your at-visit value, then you increase actually the variance. And this can be seen that your necessary sample size is much higher than actually needed. And the ANCOVA, when you use an ANCOVA, then you have the best of the two worlds. Then this is adjusted for, and you have this always lower necessary sample size. And only when you have a correlation of about 0.8, 0.9 between baseline and at-visit value, then ANCOVA and the analysis of change from baseline is approximately the same sample size needed. So often we have, it's not really clear where the standard deviation comes from. Sometimes this was done a long time ago, before the trial actually started, and when it's not really well documented. And if you have a doubt about your correlation between the baseline and your final, your at-visit value, then blinded sample size re-estimation might be actually really good idea. So for this, we developed this application. I wanted to give a quick demo. Where is it? No, this is so. Okay, so this is the application. It is it's not shared here. Why not? Just wait a second. I need to go out of the presentation probably. So now, yeah, doesn't show it. Yeah, good. So this is the application is works locally here, but it's already online, as you saw. Currently on my own Shiny Apps EO account, but later we will put this on Cytel's account. So you see here the monitor on the right side, the subjects with the endpoint on the x-axis and the estimated sample size on the y-axis, and the gray zone is where you would stop recruitment. So usually in a clinical trial, you would set minimum sample size. Below that, you wouldn't stop, even if it seems that you have enough information. And you also specify a maximum sample size. So here I prepared a situation with planned 300 sample size. Let's put the maximum sample size to 450, and then you see the monitor obviously adapts. We had here for this test clinical trial, we had an estimated treatment effect of 0.4. Planned power was 90%, significance level 2.5%, and then we can upload some test data here. We just wanted to show. Then the variables can be specified. What is the outcome variable? And then you can see what is the evolution over the trial so far of the estimated sample size. And you can also specify the baseline variables and any covariables if there are. And then we can here see now at this moment, this is just the analysis or of the at-visit value. And then we can look at this, what does this change when we look at the change from baseline? And here you saw that this doesn't change much, and we are actually in a situation where the baseline and the at-visit value have a correlation of around 0.5. So there is no gain actually in using the at-visit value or the change from baseline. This is where the curves crossed at a correlation of 0.5. But when you look at the ANCOVA, here you actually see that it seems to be that we are reaching the gray zone, so the zone where we could stop recruitment, where we already have sufficient information to finish the trial. The sample size could be enough. And that's essentially what the application does. It gives an indication of where you are with your data. And we have here this adjustment. When we have no adjustment, you see this changes a little bit, but in this sample data, it's not much of an impact for the final analysis. You also can have different parameters here. You can look at the subjects recruited instead of the subject with endpoint when you enter that you have patients in the pipeline. So for example, and then you see you're even deeper in the recruitment, stop recruitment zone because you already are above the necessary sample size with your number of patients in the pipeline. Another possibility is to look instead of the estimated sample size on the x-axis, on the y-axis, on the power. You have a similar presentation here now. What the power did evolve throughout the trial with your sample size. And that's all what I wanted to tell you. Thank you very much.
Yeah, so next is the panel discussion. And I think probably my slides didn't come through. So anyway, it's not a big deal because I just kind of had a few very generic questions noted. And I think I'm perhaps should say that I don't see really myself as a developer. I'm working as a statistician at J&J. So obviously I have to do with clinical decisions, but I was never too much into code development, but see myself as a user. I had the joy and privilege to share my office during the PhD with two people who have a much deeper understanding of R and are true developers. But still, obviously, we interact, we need to engage with each other, and there are a few or a bunch of questions and things where we should have a discussion, and perhaps even more so than we have at the moment. So also, I think even distinguishing between developing and applying shouldn't be too strict. There should always be some overlap as well with the end users, if there are statisticians or clinical scientists, doesn't matter. So when I looked at the presentations, what struck me is basically the question, what is the key advantage which makes your solution or package unique? And how much do you think about this question before you start to build a package or Shiny app which you expect to be used by others? And I mean, how important is this anyway to you? I mean, perhaps your package is just a side product of what you do anyways. So at the third time you had the same decision or problem, you decided perhaps just to package your code and then make it available. Related to that then, obviously, how does your package fit into the landscape of existing R packages and solutions? Because as shown, like, growth is still exponential, and it starts to be harder and harder to navigate which packages do I really need. And someone mentioned trust. I mean, obviously, I want to trust the developers, but there needs to be perhaps something more to build trust. So in the end, I would summarize this question or like the overall topic around stakeholders. How, who are your stakeholders, and how do you reach them? And how much effort do you undertake to really understand what the needs of your stakeholders are? And these are very generic questions. Obviously, this session is in the context of decision-making in clinical trials. So I would like to ask the panelists to come to the stage, and perhaps we can start a discussion and remind them to bring these generic questions back to the actual topic of clinical trials and decision-making in clinical programs. So perhaps, Francois, we can start with your application, because what I find interesting, it's very different from what Orla presented. So who are your stakeholders, and who do you want to use your package?
So yeah, thanks a lot. Indeed, I think this package is dedicated to probably a niche of users, which actually is not necessarily only statisticians. So to be exhaustive, I would say that, of course, it's targeting statisticians who will have to, or who want to use this type of joint models to deliver predictions, in particular, of overall survival based on changes in tumor size. This application can also be used by other groups handling these type of models, like pharmacometricians. And ultimately, the goal is to bring results to clinicians. So actually, it's targeting quite a broad range of potential stakeholders. And that's one of the challenges, because you want, in the end, to have statisticians / pharmacometricians being confident. It's coming back to the trust aspect, that the package is doing what it's supposed to do. And at the same time, you want to deliver results in a way that can be easily used or actionated by the clinical team. So we have to play on both sides. That's actually an important aspect.
And how do you reach people?
So for the technical aspect, I think it's important to to showcase examples, of course, to run some pilots, and to use reference data set, because that's what most R packages are doing. They use reference data sets that people are familiar with to illustrate the fact that we obtain the same results using this new package than what has been done in the past. So that's for I would say the building trust for the technical users. For the clinicians, it's more about communicating the results in a way that is easy to relate to. And for that, either you use classical type of display, so it's mainly visual outputs, or you have to present new displays, new results, new type of outputs, but you have to explain, and that's of course much more of a long process, or it's more involved in terms of explaining.
Thank you, Orla. I mean, if I look at what you presented, it seems so exhaustive, and it seems like quite a big project. I guess you probably have spent a lot of time, perhaps you can say something about that, to really understand and shape the product before you even start.
Mm-hmm.
But what I would also be interested in, how do you then measure from here onwards how your probability of success estimation is going to be used?
Yeah, maybe it was a little bit more straightforward for this case, because it's kind of a part of the working process, so it's something that someone does at some point in their study. And I felt like that made things so much easier, because it was going to be done. It wasn't like I had to convince people, so all that hard work was already gone before me. Whereas in other cases, when we build apps, we're saying, hey, this might help you, and either people are busy, they don't have time, or you get a very biased sample who come to you. It's the people who are the early adopters, right? They're going to join your focus group, and that's absolutely very valuable. But what I sometimes struggle with is, how do I get the median of my group? Where are the kind of in-between people, or the late adopters? How do I get them into the fold, right? Hopefully you generate enough interest and people are saying, this helped me, you generate more evidence and you build a bit of momentum, but it's not straightforward, right? And you have, as I said, the worry is you have a biased sample of feedback on your app, that when your other user group who might think quite differently come, they're like, oh, what is this? So it's always very difficult. In terms of impact, I think specifically for POS, probably what we need to do now is look back retrospectively. It's always very hard to predict the future, and I think certain disease areas are harder or easier than others. So again, I think making sure that we're kind of healthily skeptical about what we're doing. We don't forget that everything is some kind of estimate, and we challenge it and we go back. Also, there will be other people who publish different things. We need to keep the mindset of, just because we've done this ourselves, what does another method look like? How does it compare to this? And are we keeping going with ours? Are we changing? Are we adapting? So it's kind of that, I don't know, strong beliefs loosely held type of principle, right? That we always keep critical and keep changing what we're doing and adapting, I hope.
Yeah, Ursula, what I would be interested to hear as well is perhaps your thoughts on, I mean, clinical trials and programs and decisions are so manifold. But I mean, there are different requirements for certain decisions, right? There are pre-specified decisions you cannot negotiate. And others you can perhaps be a little bit more flexible. If you talk to your stakeholders about an R package or Shiny app, do you discuss this at all? The requirements, technical requirements around what type of decisions you're going to address?
The definition of stakeholders now becomes, it's very different in different projects. So when we talk with our clients, then we are very clear on which tool you use for what, in which trial. So this tool, as it stands now, is a communication tool. It's not a decision tool as it stands now. It is not a decision tool for a phase 3 trial, but it can very well be a decision tool in a phase 1 trial, because one of my original thoughts behind it was coming from a trial where I was being asked after every 4 patients how we do about the variance. And I think having such a tool and showing them already in the beginning that when you have very few patients, your confidence interval for your sample size is still very high. So in that respect, it's a communication and a didactic tool to show people what a constant question on how do we do in the variance, what that actually means. And so I have these different situations in mind. And in a phase 3 trial, this would be a communication tool after, in parallel, you would have the programs that are for the pre-specified analysis. But still, this would enable the sponsor, which are always very nervous, to have this feeling about where they are. And it doesn't cost anything because it's blinded sample size re-estimation. And of course, we need some discussion and some, we have to show that this doesn't harm type 1 error and that this trial data integrity by the data flow that we have has to be protected and data integrity has to be kept. So, and all that will always be discussed.
So you would shape it completely different if it would be used for an IDMC compared to what was presented here, but it's very much linked.
It's very much linked. The basic tool is the same, but how you implement it will be done differently depending on whether you are in a confirmatory pivotal trial or whether you are in a phase 1 trial.
So then let me ask again, what would you add if you would make it an IDMC tool?
What I would add is in the background, actually a second implementation, and second independent implementation that does run the same calculations.
Good, thank you. Perhaps at this stage we can open up to the audience if you have any questions.
Yeah, I only have a quick question for François. And I was wondering, how did you decide to create a new package instead of contributing to an already existing package? Anyone else, of course, is also free to answer.
When we were looking into this, we haven't found anything that would, on which we could contribute or on which we could build. And that's how I decide. And that is an arbitrary process because really looking for what is already existing is horrible. I would have used something else if there was already something else on which I could contribute.
Thank you.
Yeah, for GM Post, it was probably a bit the same. We checked whether anything was available for this very specific question. There was nothing. We did not went into too much details, I have to say, in checking whether we could have extended some of the previous packages because they were starting from different language. It was not necessarily always Stan in the background. And actually Stan is one of these, as one of these unique feature that it can also encode ODEs, ordinary differential equations. And that's one of the feature we were also interested in. So probably it would have been even a larger effort to restart from something existing already. And that's what was the motivation for starting from scratch.
This question is more so for Orla, and congrats on getting to the finish line of your product. I could feel the blood, sweat, and tears, so congrats. My question is around, can you show us like a concrete example of how a team has used this, and who is your main audience? I imagine it's statisticians, and what endpoints did they explore if that information is not classified?
Yeah, absolutely. Thank you for your question. So who is our main stakeholder is usually the biostats lead on the study. And then in some cases, if they have the data that they need to run the calculation kind of as is, they can work relatively independently. In some cases, we do some workshops on elicitation to try and understand, particularly if the endpoints in phase 2 are very different to the endpoints in phase 3, we need to understand how to link those two entities, and that might take some form of elicitation with experts in the room, and that is something that the team that we would facilitate, for example. I think we have supported, so we have a usage metrics app as well. It's probably a little more optimistic because everyone going in doing a sample calculation is counted. So I took some off, but around 70 studies we think we have supported to date. And what they would be doing then is taking their POS metrics at different points along those steps and using that as part of the decision when they go and present their study and go to the decision makers around whether it will go to phase 3 or otherwise. So I think it's been part of that pack of information, and it's mandated, so everybody does it, right? I think, again, as I said earlier, the really harder part is like when it's a choice and you're trying to get adoption and uptake and sustain and change management. It's much, much easier when someone comes to you because they're like, okay, I got to get this done. Let's work on it, right? And then you're in a privileged position to kind of support rather than convince.
Also a question to Orla. So looking at your presentation, I had the feeling that you put a lot of effort into software design. I've seen, I think you tried to do a loosely coupled architecture with API and so on, and then CI/CD with a high degree of automated testing.
Yes.
Can you maybe speak a little bit to, that was a lot of effort. Do you get a payback, and how is that? How is your experience with that?
Yeah, and I think we all came to the project at a different point in our, let's say, software development evolution. So I've been working in Python for quite a while, so I had been using R. So I think, and then some of us had been in R for many years. We have David Grandjean, who's written books on Shiny. So we were all, as well, at really different points in our mindset. So what we did at the start is we said, like, what is our criteria for the definition of done? And that was a really important thing. So is it code reviewed? Have you written the tests? So that no one could say, my task is done, it's Friday, woohoo, unless all of those things were met. And we as a team committed to that. And then we also committed to being like, Orla, it's not done yet. Oh, okay, fair enough. Like we committed to kind of calling each other out on it. We also tried to commit to pretty rapid code review. So I think what's very difficult is you're saying we're going to code review, but you're waiting days for it. So we tried to think about planning our weeks as well. And the other thing that we did is to our managers, we said, hey, this is important. We want a week of doing absolutely nothing but this. We're at a really crucial point. And at the end, we're going to try and commit to you that this feature is finished. So it took discipline. It took a bit of talking. I think we had a lot of support we didn't really need to convince our managers. I think they were like, if you think it's the best way, that's fine. Build it into your timelines. And where then it paid dividends was at the end where it got faster and we're developing more rapidly. What I said earlier about committing your changes and then your whole pipeline failing, as much as I hated it, I was like, okay, it's good to know. So that was really, really valuable. And then we were using this as examples now in our team to say, if you do this up front, look at the way this failed, we wouldn't have known. And even now we're finding, like, it's not that it's perfect, we still find really subtle bugs in our code, so I don't want to give the impression that everything we have done is completely flawless, but the really big things, I think, and that we found up front. So yeah, we had to have the appetite early and then also be open-minded to each other's ways of working, and then also at the end be like, okay, yeah, it's good that we did this, but let's communicate to our colleagues so that we can enforce this as more of our way of working as well. So yeah.
Yeah, so perhaps just coming back to questions we have discussed before as well. I mean, especially if you look at Shiny apps, it offers a lot of additional interactivity. But I mean, we're taking decisions and they have far-reaching consequences in clinical programs. And my question would be, so do you think, you offer a lot of freedom to the end user, right? But do you give any guidance on how often you should take these decisions? I mean, you basically allow everyone to take a look at the data and change their opinion every day. Any thoughts on that? And perhaps related to that, you give a tool which produces a lot of output, but basically we should also document decisions. So the whole archiving, do you have any thoughts on that?
I have a lot of thoughts on the question of what do we allow the user, and what do we discuss. I'm started with the pharma industry only in 2017, calling Roche Diagnostics previously not as pharma, and Unilever certainly not pharma. And democratizing statistics is something important in my eyes. That means that I think, especially in the early phases of clinical trial decision making, which is also that, most of the information or most of the knowledge is in the heads of our fellow scientists. And the communication means that that we have to translate our statistics into something that they understand, and they need to understand our statistics, and these tools are enabling, and discussing things is not harmful but good for decision making. So, and when it comes to the confirmatory trials, if people have seen these type of thinking previously, they will also understand what the confirmatory means. So my strong opinion is it's not dangerous to let the user use things. You have to give them training, you have to be at the side of them, but actually the decision making will become better the better we communicate with our fellow scientists.
Yeah, I think maybe part of what we were doing was kind of scary. So we were building an app or a package that was going to give someone a number that may or may not affect the decision as to whether a trial goes ahead. I'm not naively saying you just put the number in and it's decided, but I found that really scary. And I thought, I'm in this privileged position of, you know, working on a tool, and I go home and I come back the next day and I work on the tool again. But like, if you're working on a program or a study for so many years and you're at this point of calculating this probability of success that's going to determine if something that you're so invested in goes forward, I think you also have to think about what state of mind people are in. And so that's why building these good practices in was just a no-brainer, because we needed to ourselves be really sure that what we were doing was as rigid and robust as possible. And then we also generate a lot of artifacts. So reporting, we do a lot of sensitivity analyses in the report of if it had been this, what would it look like if it had been this, if you took this out, if you had no industry benchmark. So there's a ton of information that we hope helps to support people in the kind of understanding and confidence in the method, even at that more kind of user experience where you're not an expert in it. So I think those were the things that we did, which was important. But as I said, I still find it a little bit intimidating that we generate this number sometimes. So yeah, always mindful that we need to be super careful about it.
I will give a short answer for GM Post. We don't have a shiny app at the moment associated with it, so that's the easy answer. To come back to what we said initially, we are still in the process of trying to explain the outputs in a way that is easy to digest for the clinical team. I mean, these models, even though they may appear quite simple, they aren't actually. So there is a lot of learnings actually on our side as well on how to present these models so that they can be used efficiently. So not there yet.
Thank you. Any questions from the audience? If not, then I think let's thank the speakers again, and then we will close this session and have a coffee.